In this lecture, we will discuss capillary electrophoresis. So, capillary electrophoresis is a collection of techniques by which we can separate ionic species from a mixture based on the distinct mobilities of these ions. And we have seen earlier that for small ions, for example, the mobility depends upon the charge and the size of the ions. So because different ions will have different charge and size, they will have distinct mobilities, and therefore we can separate them based on the mobilities, because these different ions will migrate at different velocities due to their dislight mobility under an applied electric field. And separation of these ions or ionic species from a mixture is important in analytical chemistry, because let's say if we consider tap water or milk or any other fluid which is or any other solution that is consisting of different ionic species, and we need to both identify as well as quantify the amount of different ionic species present in a sample, then the first step is that we should take that sample and separate it into distinct parts, the individual parts corresponding to individual ionic species, and then we can quantify what is the concentration in the original sample. So in this lecture we'll look at a specific technique of capillary electrophoresis, which is called the capillary zone electrophoresis. Which is also called CZE. So there are different electrophoresis techniques, and there are both linear and non-linear techniques, but the simplest one is the capillary zone electrophoresis, which is actually a linear electrophoresis technique, and we'll see what do we mean by a linear technique. So this is the typical experiment that one would do for a capillary zone electrophoresis process. So what we have over here is we have a capillary which is of the radius 10 to 100 micrometers. In usual capillary electrophoresis, in conventional electrophoresis, the length can be quite long. It can be 50 centimeters to 1 meter, but in microfluidic zone electrophoresis we can shrink down this length as well, but the process remains the same, so we'll just focus on the conventional zone electrophoresis at this moment. So in this capillary, which is of the order 10 to 100 micrometers in diameter, we fill the capillary with a background electrolyte. So what is a background electrolyte? Background electrolyte is a mixture of an acid and base, that is a mixture of positive and negatively charged species, and such that, and usually we choose background electrolyte which is a buffer. That means it which is a buffer which tries to keep the pH fixed. So motion of these ions does not alter the pH of the system. So you can either make this electrolyte buffer by having a mixture of strong acid plus weak base, and otherwise you can have a weak acid plus strong base. So for example, you can create a, a buffer by having let's say X concentration of sodium hydroxide and double concentration of let's say acetic acid, and that will be a buffer solution. So buffer is needed so that the pH does not change throughout the experiment. Now let's say that our sample mixture is consisting of let's say sodium and potassium ions. So we have a mixture of sodium and potassium ions, and we need to first detect these ions and then also quantify their concentrations. So what we do is that we dispense small amounts of sodium, the mixture, which is in this case composed of sodium and potassium ions, such that the concentration of the sample that we want to analyze is significantly smaller than the concentration of the background electrolyte. And this condition is important for zone electrophoresis, because when these concentration of the sample ions is small, the overall conductivity of the solution at every location along the channel does not change. It is governed primarily by the background electrolyte. So at every point the electric field is constant, and the motion of these ions or sample ions does not affect the electric field, and we'll see that this leads to a linear process. So that means the speed of this ions, which is mobility of ith species times the electric field, remains constant throughout the process, and that's why we choose high concentration of background electrolyte. Otherwise the process can become non-linear. So what happens is that we start with two overlapping peaks of concentration of sodium and potassium in this case, and when we apply an axial electric field by applying voltage across the two ends of the capillary, so these two species, they start migrating under the effect of the electric field, and the migration velocity is given by mobility times the electric field. Therefore these two peaks will separate out, because the species with high mobility will overtake this or will move ahead, and the species with low mobility will remain behind. Along with this differential migration, these peaks also diffuse. Okay, so over time these peaks will also diffuse and broaden. So at the end, what we do is that we put some kind of a detector over here, and when these peaks migrate past these detector, we can record the signal, and we can identify the peaks, and we can also quantify the concentration. So to understand or to model this process, for the moment we'll assume that there is no electro-osmotic flow. So let's say if this is a glass capillary, we would expect an electro-osmotic flow along the applied electric field, but we can always apply some kind of a coating of the inner surface and suppress the electro-osmotic flow. So just to understand the process, we'll assume that there is no EOF at the moment. Now to model the motion or the transport of these ions, what we'll do is that we'll go back to our Nernst-Planck equations, and for species 1 and 2, so if I is 1 and 2, the Nernst-Planck equations, in absence of any bulk flow, simplify to this. So here we are assuming that the process is one-dimensional, so we have just written one-dimensional transient equation. Now, if electric field is constant and uniform throughout the channel, then E is a constant, and we also for this analysis assume that the mobility and diffusivities are constant. For strong acids and bases, so in this case, let's say for NaOH, the strong base is fully ionized, so the mobility for fully ionized ions is constant, and diffusivity also depends on mobility. So in our case, we'll say that mobility is also constant and electric field is also a constant, so we can simplify this these two equations as this. So these two equations are for species 1 and species 2 over here. Now what is this equation? This equation resembles our usual advection diffusion equation, but here instead of advection we have electromigration, and the speed of migration is dependent upon the mobility. So now we can actually see that why these two zones migrate at different speeds, because the speed or the advection speed depends upon the mobility, but these species also, the peaks also diffuse because of the diffusive fluxes. So what happens is that if you have a detector over here, so you don't need to visualize the whole channel, you can certainly do that, but even if you have a point detector, these peaks will migrate over this detector, and finally you can record a signal where you have, let's say, signal on the vertical axis and time on the horizontal axis, and the typical signal would look something like, so for two species we'll just have two peaks, and the fastest ion, the faster of these two ions, in our case actually potassium is a faster ion, will reach this detector at smaller time, and the slower ion will reach at a later time. So by noting this migration time, you can actually find which peak corresponds to which ion. And you can also do it by simple calibration. So let's say you have a sample which is composed of potassium and sodium, and if you really want to identify which peak corresponds to sodium or potassium, what you can do is that you can spike the sample with an additional amount of potassium, and you'll see that this peak will grow, and then you can say that certainly the peak which comes at this migration time corresponds to potassium. Similarly, by adding known amounts of the sample, you can find the area under this peak and correlate that with the initial concentration. So the area under the peak can be used to evaluate the quantity of this ion, or the concentration of this ion in the initial sample. So in this way we can both identify, based on the location of the peak in this signal, that which ion is present, and we can also quantify its concentration. And there are many ways of detecting these peaks. There is no single way, so it depends upon the sample to sample. So you can use any method like UV absorbance, conductivity detection, or fluorescence detection. So in some cases you have these ionic species which are autofluorescent, so they emit fluorescence, or you can tag these species with a fluorescent molecule and you can detect based on fluorescence. So in microfluidics we usually use fluorescence based detection because it is more sensitive. Now in practice you need not suppress electroosmotic flow. Actually electroosmotic flow can be beneficial. So let's say that in this example we also had some other ion. So let's say we had the sulfate ion in the sample as well. Now if we have a glass capillary and we apply electric field towards the right hand side, then the sulfate ion would move towards the left. Okay, so one would expect that we won't be able to detect the sulfate ion, but usually the electro-osmotic mobility is quite high, and even though this sulfate ion wants to migrate in this direction, the EOF mobility is so high that it will again lead to a fluid flow, which will again advect this sample, even negatively charged sample, towards the detector. So within the same using the same system, in the same experiment, you can detect both positive and negative ions, provided that the magnitude of EOF mobility is larger than the magnitude of the ion that you wish to detect. Now, having looked at the basics of capillary zone electrophoresis, we can now look at some simple scaling of what are the conditions that will actually help us in obtaining better separation. So one thing that we can see from this figure is that these two peaks can be clearly identified if the distance between these two peaks is larger than the peak width. So there is a competition between the relative migration velocities and the diffusion. So if these peaks are not moving, have not moved significantly further apart, and there is a lot of diffusion, then we won't be able to clearly detect the peaks. So what we want is that these peaks should be separated by a distance which is much larger than the diffusion length of these peaks. Now to model that, what we can do is that, so let's say that, so we look at this particular case. So if we have concentration versus the axial coordinate, so let's say at time t, the relative the distance between these two peaks, or the peak to peak distance, would be, so the relative mobility of these two ions, so mu1 minus mu2, where 1 is the fastest ion and 2 is the slower ion, times the electric field, is the relative velocity of these two peaks, and times time t is the distance by which these two peaks are separated over time t. Now what we want is that this distance should be significantly larger than the diffusion length or the width of these two peaks. So if we say that we start out with a really sharp band of these two sample ions, we see that the width of these two peaks scales as the square root of diffusivity times the time. So sigma 2 also scales as diffusivity times time, and the diffusivity is of the second species. So we can define a metric called resolution, and the resolution, let's say R, is defined as the distance between the two peaks divided by a measure of the width of these two peaks. So that is given by square root of sigma 1 squared plus sigma 2 squared. Now we can simplify this relation, and let's see what we get. So we have delta L is mu1 minus mu2 times electric field times time, and this is d1 plus d2, which is the diffusivity times time and the square root of this whole quantity. We can further simplify this as mu1 minus mu2 over d1 plus d2 times electric field, and now we have square root of time. So what is the typical time that this electrophoresis experiment will take? So we can say that the typical time taken is the length of the capillary and divided by the speed of the fastest ion when the first peak actually reaches the detector. That is the typical time. So if you substitute this relation in this equation for resolution, we get mu1 minus mu2 over square root of d1 plus d2 times L times electric field over mu1. Okay, so what does this relation mean? This means that if we take a longer capillary, then the resolution would be higher, and if we take higher electric field, then the resolution will be also higher. Okay, so that's because if you take higher electric field, then you can have faster separation and the peaks will have less time to diffuse, so you will get better resolution. But actually electric field is the voltage drop over the distance. So this quantity is actually delta V. So what matters is that how much voltage drop you are applying across the channel, and also if you have larger mobility difference, then you can get better separation, and that's obvious because the peaks would move at a relatively higher relative velocity. So one thing that is clear over here is that if you need better separation, you need higher voltage drop, and typically we use very high voltage drops in our voltages for electrophoresis. So in conventional electrophoresis, where the channel length is about 1 meter, the voltages that we use are of the order 10 kilovolts. So we need to apply about the electric field of about 10 kilovolts per meter to get good separations. So this is a brief introduction to capillary zone electrophoresis. Now we haven't discussed about how microfluidics can help us in achieving better electrophoresis, and that we will discuss in the next lecture. In particular, we will see that what are the advantages of miniaturizing this system and making a microfluidic system in achieving a proper analysis. And one thing is clear is that if you reduce the length of the channel, you can do the analysis at a faster speed. So that is the one of the advantages that you get by miniaturizing this electrophoretic system.